everyone, and welcome to chapter 3.3. This is factors affecting enzymatic reactions. Say that really fast and it's a tongue twister, I'm telling you. Anyways, in this particular video, we'll be learning 4 different factors that affect the rate of reaction. These are the 4. There is a 5th thing that affects the rate of reaction, and we're going to cover that in our next lecture, okay? So not this one. Let's start with the first one really quickly, shall we? Substrate concentration. Now, let's recap and think of what we actually have learned in the previous video. We learned about a catalyst reaction that catalyzed the breakdown of H2O2 to water and oxygen, and then we collected those that oxygen over time, and we can plot a graph, right? So here, oxygen is actually a product. This is actually a graph of product concentration against time. The x and y axes on graphs are incredibly important. Okay, remember that the x-axis is always independent variable, in this case time, whereas the y-axis, right, in this case product concentration, is the dependent variable. So this is what we are manipulating, and this is what we are measuring. But anyways, we can see here that there is another factor involved. One experiment, they did it at a lower substrate concentration. In another experiment, they use a higher substrate concentration. What do you observe here? Now, you will realize that the shape of the curve is pretty much the same. It looks like that the volume of oxygen increased for both of them and then tailed off. Okay, so the volume of oxygen increase, and then it stopped increasing. So at this point, basically, the rate is 0. The reaction has stopped because the substrate has run out. So this is true for both of them. Now, what is different is, well, the product concentration in the end. So if you have more substrate, you have more product concentration. That's one. But another very interesting note is the initial rate of reaction. So how do we calculate the initial rate of reaction? So this is a reminder for you. Take the gradient of the tangent at 0 seconds. So this is the gradient of the tangent. And you can see here the steepness of the graph, right? So for higher substrate concentration, you can see that the graph is steeper. The gradient is more, the line is steeper. So more steep means higher rate, whereas lower substrate concentration has a lower initial rate of reaction. We find that the rate of reaction is higher for the reaction with a higher substrate concentration. You can write that in multiple ways. Anyways, take this experiment, okay, and do it with a larger range of substrate concentration. Okay, do many substrate concentrations, so you get many curves that go like this, right? And then you take the initial rate of reaction of every single curve. So you basically do multiple experiments of multiple different substrate concentration, and then you measure the rate of reaction of each one of them, the initial rate of reaction. And I promise you, you will get a graph that looks like this. Now, it looks like it's the same shape, but it's not. It is not the same. It doesn't mean the same thing. It's the same shape, fine. It doesn't mean the same thing. How do we know? Read the axis. Okay, x-axis is always the independent variable. This is what we manipulate, what we change. Rate of reaction, y-axis, is our dependent variable. This is what we measure. This is a graph that is derived from many different experiments. So we can see here that as the substrate concentration increases, so increase in substrate concentration causes increase of rate of reaction. So increases, but until a certain point, the rate increases and then the rate plateaus. Okay. Now, at this point, right, the rate is not 0. I think that's a very common hypothesis, right? It's because you're thinking of this previous graph right here. You're like, oh, look, the reaction has ended and therefore it plateaued. But this x-axis is time. This x-axis is substrate concentration, and the y-axis is rate of reaction. So at this point here, at this line here, rate is high, is at the highest. Okay, and it's just not increasing. It's the highest though, it's just not increasing. That's all. All right, so I think to break this down and to explain it, it's best to, like your answers for graphs, split the graph into various sections. So we're going to split the graph somewhere here, vaguely here. We're going to split it into low substrate concentration and high substrate concentration. Let's start with what happens at low substrate concentrations. So at low substrate concentrations, there are some active sites available for binding and not all are occupied. So some enzymes are bound to substrate, some are not. And because of the low substrate concentration, there are only a few collisions between enzyme and substrate. And therefore, less substrates bind with an active site and fewer enzyme substrate complexes are formed. Overall, we say that the substrate concentration is limiting. It is because of the quantity of substrate, right? It's the concentration of substrate that is keeping the rate from increasing. Now, what happens when you increase the substrate concentration a little bit more? Well, the rate will increase because more active sites are occupied as substrate concentration increases. And therefore, at low substrate concentrations, rate of reaction is proportional to substrate concentration. You can see in this previous graph right here, okay, you can almost draw a straight line here at very low substrate concentrations. It's almost a straight line. Rate is almost proportional. Now you can see here that rate slows down. It's still increasing, but the rate of it increasing is slowed. Okay, and then it plateaus. Right, so what does that mean? Why does it plateau? So at high substrate concentration, this is what happens, okay? The rate increases to a plateau, and we call that plateau a Vmax. I'll talk about that later. Vmax is the maximum rate of enzymatic reaction. Again, I'll talk about Vmax a lot more later. Now, what happens at Vmax? What happens at this maximum rate? That's high substrate concentration, right? So basically, it doesn't matter if you increase the substrate concentration. The enzymes there's only so many enzymes to go around. So it's going to only process, say, like 7 at a time in this example, right? So 7 enzymes are going to function at one time. They are occupied, they are busy. Even if you add a lot more substrate, like a lot more, no matter how much you add, there's only 7 enzymes here, and therefore it can only process 7 at a time. At this point, at Vmax, we say that all active sites are saturated. Okay, the enzymes are in full capacity. All active sites are saturated. Maximum number of enzyme substrate complexes are formed. What is limiting, right? What is limiting the rate of reaction from increasing is the enzyme concentration. This is the factor that is keeping the rate from increasing further. Further increase of substrate concentration will not increase rate, as we see here on this side of the screen here, right? You can increase it a lot more, and it's not going to increase the rate of reaction. It's just going to process 7 at a time. And this is why the rate plateaus. Now, before I move on and talk a bit more about Vmax and Km, which is the next thing I'm going to talk about, don't worry about it first. I just want to bring your attention to the blue color words. The blue color words on the slides of this chapter are recurring words that should appear in every answer you write in your structured questions. Yeah, active sites, binding of substrate to the active site, enzyme-substrate complex, collisions, limiting factor. These keywords should always appear in your answer. Okay, you will see more of them as you go along. For now, let's go into Vmax and Km. Now, we have established that at low substrate concentration, some active sites are available. That's because at low substrate concentration, well, there's lesser substrate. So substrate concentration is the limiting factor here. But at high substrate concentration, okay, especially at Vmax, all active sites are saturated. All active sites of the enzymes are saturated. And the enzyme concentration is the one that's limiting the rate of reaction from going up, from increasing. So what do we mean by Vmax? Vmax, okay, as we mentioned just now, is the maximum rate of reaction. Only applicable to this graph here, only applicable to rate of reaction against substrate concentration graph, okay? So what is V? V is velocity, which is rate to us. Max is maximum, basically, obviously. So Vmax, maximum rate of reaction. Now, why is it useful? So, we can take Vmax, this value, and we actually can calculate another value called Km, which is useful to us. I'll explain why it's useful in a moment. This is how you get Km. At Vmax, it is the maximum rate of reaction. Okay, again, it only applies to this graph of rate of reaction against substrate concentration. Okay, at this level, Vmax, we're going to find half Vmax. So that's around here. Am I right? So we're going to take half Vmax and we're going to read the substrate concentration from the x-axis. So draw one line down and read the substrate concentration. Then we'll get this value known as Km, or the Michaelis-Menten constant. Now, what is it? Let's define it a little bit. So, as we said just now, Km is the substrate concentration at half Vmax, right? So this means that Km is the substrate concentration at which the enzyme works at half its maximum rate, right? Vmax, enzyme works at half its maximum rate. Makes sense, right? Now, at Vmax, active sites are fully occupied, but at Km, half the active sites are occupied by substrate. So because it's half Vmax, right? So at Km, half the active sites are occupied by substrate. And why is this helpful? Why is it helpful to know this value? Now this is because Km is a measure of enzyme affinity. What do we mean by affinity? Affinity is the degree of attraction between molecules. So here's a little example. Let's think of attraction in terms of human beings. I find that quite helpful. So if this is a boy here and that's a girl, and they somehow are very compatible to each other, and you know, if they're compatible and they, you know, the attraction is there, the attraction is strong, if you might say, the affinity is strong, then it would take a very small incident or very little effort in order for a relationship to work out, correct? If the attraction is strong, there's commitment, obviously, there's emotion, it will take less effort to get there, right? Whereas if there's another person in the picture and you know that, well, the girl here may not be the most compatible for this guy. I'm not gonna say pretty, that's not nice. But yeah, not very compatible with the guy. It would take a lot more effort for this relationship to work, isn't it? Like, it can work, like a lot of relationships can work with a lot of effort, right? Every relationship is effort. So, but if you're not attracted to each other, it's gonna take even more work to build something like out of it. So it's the same with Km and affinity, right? Let me erase this. So if an enzyme and a substrate is kind of have a high affinity to each other, then very little substrate, less substrate is needed, less substrate concentration is needed in order to reach half Vmax, right? This means that the lower the Km, the higher the affinity. So it's like inversely proportional to each other. Yeah. Let's do the reverse. I find that helps some people sometimes. So let's say an enzyme, this enzyme, different substrate, right? Their degree of attraction, their affinity is not that high. Low affinity, low attraction. Now it will take a lot more substrate in order to reach half Vmax. And therefore, half Vmax. And therefore, you will expect that Km to be higher. So you can see low affinity, higher Km. So if we do an experiment and we get a graph, right, that looks like this, we can find out the affinity of the substrate, how much attraction there is, just by looking at the Km value. The higher that value is, higher Km is, lower the affinity. Now for enzymes with high Km and low affinity, this is what we use words to describe it. Okay, higher substrate concentration, as I said just now, is needed to reach half Vmax. More effort needed here. Okay, enzyme needs a higher concentration of substrate to reach Vmax. Why? Because enzyme forms fewer enzyme substrate complex in the same unit of time. And this might be because the active site is less good fit for a substrate, possibly. And therefore, the reaction is kind of harder to happen. So yeah, here's a little exercise. Now, just looking at hexokinase, which one is it more likely to bind to? Fructose or glucose? Pause here if you want to think. Okay, just pause here. Now, let's look at the Km value. Glucose, very low. Fructose, pretty high. High Km means low affinity. Low Km means high affinity. It's opposites. Therefore, hexokinase would be more likely to bind to glucose instead of fructose. Hexokinase has a higher affinity for glucose. Answer: glucose. Okay, so yeah, this can be very past year question. Now, we saw Vmax and Km just now, and we saw how to calculate it just now. But there's a problem, experimentally in real life. Too many experiments are needed for the graph to plateau in order to find Vmax and Km. Very often, you end up with a graph like this or like vaguely curving like this. You need to do many, many experiments in order to see that curve truly flatten out. You can see it slow down right here, but it might, go further. I can't draw any further than that, but it can go even further. It can be like slightly tilting for ages and then only plateau. So that is a problem. How do we derive half Vmax and Km if we don't know Vmax? Okay, obviously this is just a diagram, but how? How do we do that? Now, there's a solution, a very mathematical one. You can convert a curve to a straight line if you plot a double reciprocal graph. Plot 1 over V against 1 over substrate concentration. So V, is a rate of reaction actually, rate. So instead of plotting rate over substrate concentration, plot 1 over rate against 1 over substrate concentration. Yeah, and if you do that, this is the graph. So ta-da, you just converted curve into a straight line. But how do you find Vmax and Km from the straight line? Now, for this, you need to understand a concept. When in our previous curve, we drew rate against substrate concentration, and it kind of increased and plateaued off. Right, this plateau is when substrate concentration is very high, and you will assume that the graph will continue to plateau until substrate concentration equals to infinity, correct? So how do you find Vmax? Vmax is when substrate concentration is at infinity for sure. So how do we find Vmax on this graph? Let's find 1 over substrate concentration and 1 over Vmax. So if substrate concentration equals to infinity is Vmax, it should be 1 over substrate concentration, which is 1 over infinity, is 0. It's too small, so it's 0. So since Vmax is at substrate concentration equals infinity, 1 over Vmax is at 1 over substrate concentration equals 0, which is the y-intercept right here. So y-intercept is 1 over Vmax equals to 8.5. Convert that, Vmax equals to 0.118. Vmax at infinite substrate concentration is 0.118. Now, how do we get Km? Now, Km is here. Okay, is at 1 over V equals to 0. There is a reason why, but I don't want to explain it. It's quite mathematical, but trust me, the x-intercept is negative 1 over Km, and therefore, if you take it, make it positive, and then flip it around, it is also 0.118 here. So you get the Vmax and Km values from the y-intercept and the x-intercept. Now, if you want to know why, you can talk to me personally. Okay, so this is a simplified diagram. Again, if you didn't get any of that, how to derive it, don't worry. Just remember that in the double reciprocal plot, the y-intercept is 1 over Vmax, and the x-intercept is negative 1 over Km. You don't need to derive it, you just need to know how to use the formula. Ding! Okay, with that, we're done with substrate concentration. We can move on to our next factor that affects the rate of reaction, enzyme concentration. Here is another graph that looks the same but is actually different. Here's a graph with the same shape, but if you look at the axes, it's different. Our independent variable now is the enzyme concentration, and dependent variable is the rate. So basically, we keep our substrate concentration constant now, and instead of increasing substrate concentration, we increase the enzyme concentration in many, many different experiments, and then we plot this graph. This graph takes many, many different experiments, okay? Now, again, we can explain this graph by splitting the graph into 2 parts. What happens at low enzyme concentration? What happens at high enzyme concentration? Let's find out. So this is the enzyme working 7 at a time. If you increase it a little bit more are going to work together. So what I'm saying is this. At low enzyme concentration, and as you increase the enzyme concentration, more enzymes are present. Therefore, more active sites are available for substrate to bind. And therefore, also, because of the increase in concentration, there's increased frequency of collision. So obviously, more enzyme substrate complexes can form. And therefore, rate increases as enzyme concentration increases. Now, there is a but, right? Rate increases as enzyme concentration increases only until a certain point. Because at high enzyme concentration, the rate of reaction levels off. It's very high, but it doesn't increase anymore. It just stays that high. Why? That's because substrate concentration becomes a limiting factor. Okay, you can add as many enzymes as you want. Many enzymes as you want, but there's only so many substrates to process. So substrate concentration here is the limiting factor. Maximum number of enzyme substrate complexes form, and therefore the rate of reaction levels off. Okay, so what did we just talk about? Low enzyme concentration, less active sites available. As you increase it, there are just more active sites, and therefore enzyme concentration, rate of reaction increases. Enzyme concentration is the limiting factor. However, if you continue to increase the enzyme concentration, there would be a point where the rate levels off. Why? Because the substrate concentration is the one that's limiting. The max number of enzyme substrate complexes have formed. So yes, that's enzyme concentration. It's very easy. Next, temperature. So this is the third factor that we're going to talk about. So temperature's graph looks radically different, right? Phew, finally a graph that's a different shape. Now here is quite interesting because the rate of reaction doesn't go up and then plateau. It goes up, it goes down. What does it mean? Well, that's because at this point, no reaction. At this point, no reaction. Optimum reaction here. Okay, so how are we going to break this down? Low temperature, high temperature, and optimum temperature. You realize that the curve, that the slope here is less steep. This is much steeper, right? That is intended. Now let's see what happens at low temperature. Now at low temperature, as temperature increases from low temperatures to optimum temperatures, there's an increase in kinetic energy. So at low temperature, movement is kind of slow. At high temperature, it moves kind of fast. Therefore, it moves faster. Therefore, if it moves faster, that's going to be an increase in collisions. And therefore, substrates can bind to active site more often and more enzyme substrate complexes can form. And therefore, from low to optimum temperature, rate increases as temperature increases. Yep. Now, let's talk a bit about optimum temperature before moving on. Optimum temperature is the maximum rate of enzyme reaction. It's like the ideal temperature where the enzyme is comfortable at and best functioning at. Most human enzymes' optimum temperature is around 40 C or slightly below that. We just say in general around 40 C, yeah? But not all enzymes have this optimum temperature, okay? Now think of it as, think of the enzymes in the hot springs of, I think this is Yellowstone National Park. This is a sulfuric lake and it's a very, very hot lake. There are things living in there. So thermophilic archaea live in there. Archaea is a type of prokaryote. I don't know if you remember, but there are enzymes in those thermophilic archaea that have very high optimum temperature and they work best in those high temperature environments. The point is, different enzymes, different organisms, different optimum temperature. Not all enzymes are equal. Okay, so that's optimum temperature. What happens at high temperature, like very, very high temperature? Now, if you're thinking denaturation, you got it. So reaction actually slows down and then stops. Why? Because hydrogen bonds and ionic bonds start to break. Hydrogen bonds and ionic bonds. These are from chapter 2, guys. Chapter 2. These are the bonds that are the weaker ones in the protein's tertiary structure, okay? And these are the bonds that start to break first, causing denaturation, causing the active sites of the enzyme to change shape. And therefore, substrate cannot bind to the active sites. The enzyme is denatured at this point. And fewer enzyme substrate complexes can form, as the temperature increases. And eventually, all the enzymes are denatured and reaction stops. Yep. This is an irreversible process. Once the protein is denatured, it's denatured. Like even if you lower back the temperature, it's gone. It doesn't work anymore. So that's temperature for you. What happens? In summary, temperature at low temperature, at low temperature, as temperature increases, kinetic energy increases, more collisions, more enzyme substrate complexes. Great. This occurs up till optimum temperature. At high temperature, however, enzymes denature. Why? Because the ionic bonds and the hydrogen bonds break and the active site changes shape. And therefore, no more enzyme substrate complexes can form. And ta-da! Low rate of reaction. Now, next one is pretty similar, but slightly different. Okay, it's pH. Now you can see the graph, the low and the high pH is kind of like the same steepness, and then there's the optimum pH right in front. Note that in both temperature and pH experiments, substrate concentration and enzyme concentrations are constant. Yeah. Anyways, in this pH example, we are going to explore low and high pH in the same slide. Now, pH is the measure of concentration of hydrogen ions. Hydrogen ions, not atoms, not molecules. Hydrogen ions in a solution. Low pH means it's acidic, means there's more hydrogen ion concentration. High pH is alkaline, less hydrogen ion concentration. Now, at very low or very high pH, okay, basically not optimum, these hydrogen ions can interact with R groups of the amino acids in the enzymes. Either too little ions or too many ions can interact. And therefore, this disrupts ionic bonds and hydrogen bonds in the protein. Remember, enzymes are all globular proteins. This changes the active site shape, and therefore substrate cannot bind to the active site. Less enzyme substrate complexes form. Do you realize that these terms appear a lot? Anyways, less enzyme substrate complexes form, and therefore low rate of reaction. Now at optimum pH, okay, the right amount of hydrogen ions are there to interact with the protein while still keeping its shape. It's like perfect. It's the maximum rate of enzyme reaction at optimal pH. Beyond that, the enzyme kind of denatures. But pH is not as irreversible. Like, if you denature it a little bit because of pH, it's possible to reverse that. Temperature, though, completely irreversible. Of course, if you increase it way beyond the optimum pH, then it might be permanent damage there. But anyways, beyond the optimum pH, we said that the enzyme actually denatures. Different enzymes in your body has different optimal pH. I think we don't think about this enough. We always think of our body as kind of neutral. I don't know. And we think all the enzymes in it are neutral, but no. Different parts of your body have different conditions, different pH, and therefore these you can see these enzymes here, which are in your body, function at different parts of your digestive system. Pepsin, sucrase, amylase, arginase, they all have different pHs, optimum pHs in your body. Yep. Okay, different enzymes, different optimal pH, different enzymes, different optimal temperatures as well. So yeah, don't take it for granted. Now, what is the summary here? At very low or very high pH, that is not the optimum. Ionic and hydrogen bonds break, therefore active site change shape, therefore less enzyme substrate complexes form, and therefore low rate of reaction. The only pH where everything is fine and dandy is the optimum pH right here. And yeah, with that, we have covered 4 different factors that affect rate of reaction. In our next lecture, when I see you next time, we'll be talking about how inhibitors can affect the rate of reactions and 2 different ways inhibitors can do their job. I'll see you next video. Bye-bye!